Bacteria, as it is our topic today. So bacteria, it's when there are many. So when it is one, it is called the bacteria.
Yeah.
So now the bacteria is a single-celled microorganism. Single-celled microorganism. This microorganism, it is found everywhere on this planet Earth. Actually, everywhere you can find these bacteria. It is found everywhere. Another thing to know about these bacteria, there are millions of bacteria, but many of these bacteria, they are harmless. Many of these bacteria are harmless. In fact, some of these bacteria, they help us in other things. Like some bacteria are used to process some foods artificially, like the yogurt. Then other bacteria, they are they live on our body or in our body. Some on the skin, some inside the skin, like the Escherichia coli, which lives in the gut. The Escherichia coli, which lives in the gut, helps us helps us in the digestion, and actually, these normal floras or these they are called your microbiome because they don't do any harm to us. In most cases, or sometimes they help to prohibit the growth of the pathogenic microorganisms. So, and then others, the few ones out of the millions of the bacteria. There are the few ones are some few are pathogenic. Some cause diseases to us, so they are pathogenic. And these ones these ones they are like the Clostridium tetani, the Staphylococcus, like that. So those are examples of these the dangerous ones. Now let us proceed and talk about the classification of these microorganisms. Now, classification of bacteria. Classification of the bacteria. Bacteria are classified in many ways. We shall start with the scientific classification. The scientific classification, here we classify bacteria depending on their kingdom. Here we'll see that we shall start with their kingdom, the genus, and the species. Also the family. So this is another way of classifying the bacteria. We can classify them according to the kingdom they fall, the family, the genus, the species. So this is one way of classifying. We shall see these things in the details in our next video, in where we are going. So another way of classifying is classifying by shape. We can classify bacteria according to their shape. We have a shape. These are the shapes we have for bacteria. We have this one, majorly, and we also have this one. So this is it. You can say ball shaped or oval. Then this one you can say it's rod shaped. This is ball shaped. This is rod shaped. Majorly, these are the two shapes we have for bacteria, but some can be spirals. Some of them can appear like this. These ones, this shape is they are the spirals or spirochetes. Eh? The spirochetes. These are the main shapes we have. The third way of classifying is we have by their genetic formation. Here we can do certain tests to differentiate the nature of their genetic composition, their genome. We base on their genome to differentiate bacteria. Then the fourth way of classifying is the need for oxygen. Need for oxygen. Here we classify bacteria whether they are anaerobes or aerobes. Aerobes. Those who can't stay without oxygen, they are called aerobes. And those ones which can stay without oxygen, they are called anaerobes. So some are aerobes, some are anaerobes. So these ones can stay only in presence of oxygen. Those ones can stay without oxygen. The fifth way of classifying bacteria, we can classify bacteria according to their staining characteristics. Now the staining characteristics here, some the commonest way we can differentiate them is by use of the Grams staining. Grams staining technique. Here, we differentiate microorganisms, these bacteria, into two. We can differentiate them into gram-positive and gram-negative. And differentiate them into gram-positive and gram-negative. We shall discuss this gram staining in the future, but those ones with a thick cell wall, they are called they are the ones we call the gram positive. Then those ones with without the with a thin gelatinous structure, they are called gram negative. So here there are some examples. The first here we have the Clostridium, we have the Listeria, here we have like the current bacteria. These are some of the examples. Then here are some examples like the Klebsiella. The Klebsiella bacteria is a gram negative. Then we have the Proteus. We have the Pseudomonas. I think these are some of the examples of the gram staining. Some they are positive, some they are negative. So what we can talk about bacteria, bacteria they are prokaryotic, not eukaryotic. Prokaryotic refers to the cell, refers to a cell, which don't have a nucleus. A nucleus. You know very well this is a single-celled microorganism, the bacteria. Now, a bacterium, if it is a single cell, inside that cell, it does not have a nucleus. It only has ribosomes, cytoplasm, and the DNA, the nuclei. Inside it is a cell. Those are the only content it has. So these bacteria which cause diseases, they are dangerous to the human body. So the immunity starts to fight against them. But sometimes the immunity of the body may need support from antibiotics. So the prescriber will prescribe antibiotics to fight against these bacteria. But now, what you have to know with antibiotics, they act by destroying the cell wall of the bacteria. Or sometimes they act directly to the DNA or they act directly on the DNA of the bacteria, and they destroy it. So these antibiotics are prescribed by a health worker, but overuse of these antibiotics may result into antibiotic resistance. So you take the example of the methicillin, resistant Staphylococcus aureus. It resulted into overuse of antibiotics. So the more you use antibiotics, the more the body may develop resistance to antibiotics, or the more other bacteria can resist them, can get the ways of of resisting antibiotics. So you better use those antibiotics as prescribed by the health worker. So in our next video, we are going to look at the detailed information about the bacteria. So don't forget to subscribe. Comment your question, we shall answer it. Any question about medical, anything in the medical, just comment, we shall have to answer. So keep in touch, just subscribe. Thank you very much.